So welcome to our webinar this evening. We will be hearing about genetic diagnosis and clinical trials. I'd like to introduce our speakers, Dr. Aron Chakera, Jonathan Nguyen, and Ella West. So we will begin our webinar hearing from Ella. So Ella, over to you first.
Perfect, thank you. Now let me share my screen. Okay, perfect. So my name is Ella West and I work as a clinical genetic counsellor at RPA and a research genetic counsellor at the Garvan. And I'm just going to give a quick overview on genetic testing and counselling for ADPKD. So a few of you might know what a genetic counsellor is, but I thought I'd just quickly run over it. So we work in a lot of different spaces, but in the clinical sense, we work with people who are either themselves, their pregnancy, or their child might be at risk of or have a genetic condition. And we work with them to discuss testing, inheritance, what this might mean for them, and just see if we can generally support them in that area. I also work in the research setting at the Garvan, working in a lab where we are trying to better understand the genetic basis of kidney disease. So, quick genetics overview, promise it won't be too long. Basically, we are made up of cells, and inside of those cells, we have this thing called chromosomes. Now, we have 23 pairs of chromosomes, and we get one from mum and one from dad. Now, if you unwind this chromosome, you are going to have this thing called DNA. And all the DNA is sort of sectioned into these things called genes. Now, each gene is like a recipe for a different thing in our body. We have 20,000 genes, and we have 2 of each copy, because we get one from sperm, one from the egg, and each gene has a different function or role in our body. Now, we know that a bunch of these genes are really important in our kidneys, and if you have a change in one or both of them, you will develop cysts in your kidneys. Now, the vast majority of people, specifically with ADPKD, are going to be, it's going to be inherited from either their mum or their dad. However, in about 10% of cases, we know that their parents cannot have cysts in their kidneys, and ADPKD can begin brand new in them. Now, autosomal dominant polycystic kidney disease is typically caused by a change in either PKD1 or PKD2. There are some other genes that do cause ADPKD, but these are a lot more rare. And if you do have ADPKD, you will have a change in one copy and a working copy. Now, every time you go to have a child, 50-50, completely random, you will pass on either that working copy and you will have a child that's unaffected, or 50-50, you'll pass on that non-working copy and have a child with ADPKD. Now, I thought I'd quickly just run over autosomal recessive PKD, which is caused by changes in the PKHD1. Now, with autosomal dominant PKD, you only need one change to really cause that condition. Whereas with autosomal recessive PKD, you need both of them knocked out. Now, what this will be caused by is both of the parents being carriers, and carriers do not exhibit symptoms of that condition because they have this backup copy doing the work. But if a carrier meets a carrier and they have a child, there's a 1 in 4 chance of them having a child with ARPKD. Now, why would someone consider genetic testing? The big one is knowledge. A lot of people ask, why is this in my family? What is actually causing this? And genetic testing, with the genetic diagnosis, we can tell you and exactly what change, and we can see what gene it's in and give you a bit more information for your own knowledge. Now, I've put this in italics because a lot of the time it doesn't, but occasionally knowing your genetic change can actually give us some information about what the future might look like or what may happen in the future. Now, the big thing that I spend a lot of time talking about is what a genetic diagnosis will mean in regards to your family planning. I know that there was a previous webinar about IVF with PGT, so I won't run over too much of it, but basically a genetic diagnosis allows people to consider IVF with pre-implantation genetic testing. And what this is, very simply creating embryos and then testing those embryos for ADPKD or a genetic condition, and then only implanting those ones that do not have a condition back into the woman's uterus. Now, I made this sound very simple, but it's actually quite complicated, hence why I think there's a whole webinar on it. And the other thing genetic testing can be useful for is understanding the risk for other family members and testing other family members, because if we have a genetic diagnosis, that means siblings and other family members can get tested. A common question we get is, can my children be tested? And we don't typically test young children for AD-PKD because this is an adult onset condition. However, we see people usually when they're teens to have a chat about it, so that child can also be involved in their own conversation about genetic testing. Now, management, I'm not going to touch on too much, but one of the big things that we are talking about is gene therapies and potential therapies. Now, you'll often need, well, you will need a genetic diagnosis to be able to access these therapies. And if you've had genetic testing, getting an appointment with the genetic counselor or geneticist takes a long time. Genetic testing takes a long time. And so having a genetic diagnosis here in 2025 means that if there are therapies that you become available or in the future, you've already got that diagnosis ready to go and you don't have to go through the whole testing process. So what does genetic testing actually involve? So even though it can just be a blood test, it's a lot more complicated than other blood tests you might just get at your GP. So you'll meet with us to discuss what are you actually being tested for? What are the possible results? Because it's not just a simple yes or no. Sometimes it's a yes, sometimes it's an unclear no, and sometimes it's a maybe result. We don't actually understand what it means. Specifically for people who do not have the condition, but we are predictive testing them. There might be insurance implications, so we will discuss that. And will this change your management and how will you receive the results? We'll discuss all of that in your consent appointment. The actual genetic testing, relatively simple for you. It's a blood test or a saliva swab, and this will depend on the lab and the clinician ordering your test. The lab analysis takes a long time. Depending on the lab, depending on lots of different factors, I'm currently, for my AD-PKD patients, it's about an 8-month turnaround time. Now, this is for many reasons, because first, it's a really complicated test and they're actually reading your genetic code and seeing each change and going, is this change causing the PKD or is this just unique to you and your family? And the other thing is genetic testing, and not just for PKD, but for all genetic conditions, is getting so much more common, and therefore there's a lot more samples being sent to the lab, and there is a lot of pressure on the lab staff at the moment. So we are seeing quite a long turnaround time to get those results back. And we, as I said before, there's about 20,000 genes. Now, we don't look at all the genes. We look at only genes that are associated with kidney disease. And specifically with AD-PKD, sometimes we'll only look at genes associated with kidney cysts, not even other kidney conditions. And so that will also be discussed in your consent appointment about what we're actually testing for. And there's a Medicare item number for genetic testing. So if you have Medicare, this will not cost you anything. Now, the results, if it's a clear-cut result, you might be asking, does this result explain my features? Does more testing need to be done? Because sometimes it's not so clear and we need to test other family members or do more testing on you. Can you use this result for family planning or testing other family members? And sometimes we get a negative or uncertain result, which can be a bit complicated on our end because we need to figure out why you've tested negative or why there's an uncertain result. Now, negative results do happen in people that have really clear-cut AD-PKD. Like, we're looking at the ultrasounds, we're looking at, you're talking to us and we're like, you definitely have AD-PKD, but you've tested negative. It does not mean that you do not have a genetic change. What it means is that our technology in 2025 is not good enough to pick up that change. So that's what we're actually working on at the Garvan, is actually there's a cohort of people that have got negative genetic testing, and we're trying to better understand what we're missing, because we know they've got a change, but where is that? So that's something we're doing at the Garvan. Now, for you, how can you access genetic testing? There's a couple different options for you. You can either go through your nephrologist. Now, some nephrologists are comfortable ordering genetic tests, others are not. Some nephrologists that order genetic tests, they might actually refer you to clinical genetics to discuss the results and the family implications, and some nephrologists might not order the test and send you straight to the clinical genetics service. So there are clinical genetics services everywhere in all states. And you can come and see a genetic counsellor or a geneticist there. You just need a referral from your GP or your nephrologist. What you will do is you will meet with a genetic counsellor or a clinical geneticist, discuss your personal and family history, and you will go through the consent. Now, you can find your local service. I've just put a QR code there, and there is a link there, and you are welcome. Clinical genetics service, we get questions from all people all the time, so you're always welcome to give your local one a call and ask any questions about that. And that is me done. Hopefully I stuck to time.
That's amazing. Thank you so much, Ella. That was a brilliant overview and very practical. I know already you've hit some of the questions, particularly around the turnaround time. I think someone had a test back in January and they're still waiting for results. So that's encouraging to hear that these things do take time, as well as the fact that it's covered by Medicare. And just before we go to Jono, I was going to just put in the chat, and I'll link this later as well on our YouTube recording. We've actually had a couple of previous webinars relating specifically to genetic testing for family planning. So if you want more information on that, I'd strongly encourage you go to our YouTube channel. I'll just put it in the chat there. One was focused on Mackenzie's mission and that study done in Australia. And then also we listened to KidGen, and they had another two-part series on genetics and screening and all that side of it. So Ella, thank you. And now Jono is going to talk to us more about what he does at the Garvan Institute and how genetic diagnosis can be beneficial and the new rare disease registry. So Jono, give you a chance to put your slides up. There they are. Thank you so much. Over to you.
Thank you. All right. Evening, everyone, and thank you for having me. I'm no stranger to the PKD Australia community. However, to those I have yet to meet, my name is Jono, and I'm a researcher at the Garvan Institute based in New South Wales. And I'm focused on a study known as the Genomics of Rare Disease Registry, which you may have heard. And it's a national participant-led platform across all states and territories of Australia. And we're primarily focused on monogenic disease research. So we'll first start off with the bread and butter of the Rare Disease Registry, and that's the complete focus on understanding the disease mechanism of monogenic diseases. And by the definition, it is a disease caused by mutation in a single gene. One known examples would include cystic fibrosis, Marfan syndrome, Huntington's disease, and one that's more relevant to this community, is polycystic kidney disease. The genomic cause of these diseases are often known, however, others not as much, and ongoing research is being conducted to understand the biological mechanisms of these rare diseases. As you may know, within previous webinars, genetic testing helps identify carriers, as well as family members that are at risk across multiple generations. And that leads to the key question: what's the purpose of having a genetic test, which Ella kindly elaborated on, but as well as what's the role of genetics? So having a genomic diagnosis isn't just another word within a laboratory report. It's to patients and families inflicted with this rare disease. They're often on a diagnostic journey, often months to years, and a genomic diagnosis is the million-dollar question on what's causing their symptoms. So in turn, it really opens doors to a more holistic support that they can reach out to, therapeutic management, potential treatment, and ultimately being eligible to join clinical trials and research studies. While we are on the topic of research, there are multiple pathways genetics can ultimately help us when developing and accelerating research. And types of the developments involves the gene-based therapies, for example, gene replacement, precision medicine, which is understanding the exact mutation, allowing therapies to be tailored to an individual, and evidence building, which I'll touch on a little bit more in later slides. So the biggest takeaway and the primary aim of our study is based on how genetics really provides answers, connection, and hope to individuals and families. Because rare diseases because the other rare disease landscape, my team focuses on is predominantly genetics. Theoretically, it makes the lives of researchers and clinicians a little bit easier, as long as we identify the genomic cause of the disease. However, in reality, this is quite far from what we really experience on a case-by-case basis. As at Rare Disease Registry, we often run into a lot of problems due to the complexity and the uniqueness of each individual disease, as well as each individual family. So to throw in some quick stats in, there are more than 7,000 rare diseases affecting up to 5.9% of the Australian population. And you may have heard this before, individually with the rare diseases, they're quite rare, but collectively under a rare disease banner, it's actually quite extremely common within our community. Majority of these conditions are classified as monogenic, and in cases where you can identify an underlying genetic cause, that diagnosis, can be extremely valuable, both clinically for genetic counseling, cascade testing, and even referrals to treatments. But there are also scientific benefits as well, and these are highly tractable systems to understand the biology and disease mechanisms of these genes. And it ultimately allows us to test the potential treatments more quickly and accurately. But there are two key challenges that we've noticed and that we're currently facing at the moment. Most rare disease patients actually do not have a genetic diagnosis, and even when they do, less than 5% have an effective therapy. So from my team, we actually want to change these numbers, and by utilizing the registry, as well as adapting new technology, we hope to improve diagnostic rates, support researchers, and really collaborate with industry partners to accelerate the development of therapeutics. One of my personal goals is to place Australia on the map of research and clinical trials and to utilize our registry to connect families to these studies. One other thing that we also want to change is the way we really approach these problems. Being able to tackle rare diseases has been going on for decades, maybe even more. But rather than working within our institute or disease-specific silos, we want to sort of take a more coordinated approach to tackle all rare diseases rather than just one disease at a time. So it's a fairly chunky slide, this one, but our solution to the complexity of the rare disease landscape is our registry. And I'll try to quickly speed through this as much as I can, but some of you may be familiar with this process, as a lot of you have already signed up. But recruitment can begin through a participant-initiated pathway where you complete an expression of interest survey. The team will triage your responses and then screen for eligibility. We also have the possibility of clinicians referring directly to us as well. The registry enrollment side is a little bit more unique, where once an individual has consented to the study, we'll send through a baseline survey. And for our registry, we're not really focused on one particular disease. It would be great to pull our resources in, but because we're spread so thin across all types of rare diseases, it's quite hard to collect all the detailed information we would like. So what is being collected is what we refer to as a minimum data set, and that mainly consists of the age of onset of your disease, a genetic diagnosis, if it's known, clinical symptoms, as well as family history. We do have the ability to develop and distribute disease-specific surveys. However, using the already existing minimum database, we do already have the ability to collaborate with academia, patients, and industry. And the patients and industry section is a really big topic because we're heavily involved in the genetic diagnosis pathway of individuals, especially those that still remain undiagnosed. And for the industry partners, we collaborate a lot with biopharmaceutical companies to help establish new clinical trials in Australia, or really advise how many participants with a particular disease or gene are existing, and assist with the feasibility process of developing these studies. Almost finished, but the aims of the registry are fairly straightforward, however, very, very complex when you actually look into it. But to sort of put things in a nutshell, we do want to increase the scale of our registry, really, because although we are considered national, there's a lot of underserved communities that we haven't reached out to, and we're really trying to navigate to a lot more ethnic and minority communities that may not be represented. And we're hoping to really streamline the process of the patient journey to just make things a little bit easier. By forging partnerships with patient advocacy groups such as PKD Australia, we're really able to target the core community to understand what's needed, how we can help, as well as what we can provide to assist the communities as well. Number two is accelerated diagnosis, and I've mentioned this before. We're very passionate about diagnosing undiagnosed patients, and we're only getting better at this by generating and integrating genomic and clinical data, as well as utilizing new and advanced genomic analytic tools as well. Within the Garvan Institute, we have a huge team leveraging, and we're able to leverage on both clinical and academic expertise when it comes to patients with very, very complex genes and presentations. Last aim is to really facilitate research, and that's through a robust and comprehensive biospecimen collection. Because with biospecimens, we are able to conduct preclinical and de novo research, as well as work alongside work alongside industry partners to really help and understand the biological mechanisms of these diseases. I think that's all from my end, but this is just more of a quick overview of the team that I'm working with. And if you would like to learn more about the Rare Disease Registry and how it might be able to help yourself, your family, or the community, please don't hesitate to visit the website. I think my contact details should be on there. But yeah, otherwise, feel free to reach out to Jess, and she's more than happy to pass on my details.
Absolutely. And we have featured the Garvan Institute's Rare Disease Registry in our newsletter, and we'll continue to do so. And it's actually part of the footer, so it's always there. And Jono is very good at replying to emails. So I can guarantee if you reach out to me or him, as you've heard and as you've so well said, you're very passionate about this, and that comes across. And yeah, I'll be excited to hopefully boost that data set, and ultimately will lead to better research, which will lead to clinical trials, which segues into our next speaker, Dr. Aron, and he'll be speaking more, I guess, tying it all together really, and also speaking about the new clinical trial with PYC. So over to you, Aron. And thank you, Jono.
All right. Can you see my screen okay?
Yes, indeed.
Fantastic. Nice when the technology all comes together. So look, I'll hopefully be able to bring things home. I'm going to largely talk to my brief, which was around the PYC 003 trial, which I know a few of you had questions about, which I'll hopefully answer during this. And just also maybe provide a little bit of background context from some of the comments that we'd received prior to today as well. Because this talk, some of the information is around obviously a commercial trial that's being run at the moment. There is this disclaimer to go with things that basically says, look, we're talking about work that's in progress at the moment, and maybe don't just base all of your future investments on what I'm telling you today, but I'm very happy to answer any questions as far as I can. So the background side of things to ADPKD, I don't really need to provide too much to this audience, except to say that it's a very highly prevalent condition. And one that, probably even 10 or 15 years ago in my clinical practice, we would make a diagnosis of polycystic kidney disease largely based on either a family history or some imaging findings. And there wasn't really very much that we could do. Unfortunately, in terms of risks of progression, the data still suggests that up to half our set of patients are going to progress to end stage by the age of 60s, and treatment options have been largely limited to date. But as I'll hopefully show you over the next few slides, things are changing very rapidly in this space. So it's a pretty exciting time for what is a very common disease with a lot of different opportunities now on the horizon. Just as a little bit of background to flesh out some of the stuff that's already been there, because it will be relevant to the things I'm going to mention for some of the studies. Couple of different forms. We're normally talking about autosomal dominant polycystic kidney disease, which is the picture on the right-hand side there, where you just need one affected individual and one of the copies of the gene not to be working to develop the disease. So different to the autosomal recessive inheritance conditions that we've briefly touched on in the earlier presentations. And again, I will come back to belabor this point a little bit more, but effectively we have two copies of these genes. One's not working the way that it should do. The other one still seems to be okay. And that will link into the therapeutics that we'll discuss in a bit. The other principle that I just want to raise with people, again, as it comes to therapies and options going forwards, is this idea of homeostasis. So the body is trying to keep everything in balance all the time as much as it can. That happens at a kind of macro level. A good example is like your sort of temperature rheostat. So your body is very good at trying to keep your body temperature at around 37. If you get very cold, you'll start to shiver, generate more energy to try and warm your body up. If you get too hot, you start sweating, because there are a whole lot of processes running behind the scenes that are designed to try and keep your temperature at level. Those same processes also happen at a molecular level to try and keep everything running within the cells. So when that becomes disordered, lots of things can go wrong all at once, and that level of control that you should be having can start to become problematic. Another bit of the sort of central dogma of molecular biology, and apologies for those of you who are having nightmares back from high school or early university biology, but it is important to some of the conversations coming up, is this information flow that we go from a gene in DNA through to mRNA, which is the sort of initial product, through to a protein at the other end of things. And this, like that homeostasis stuff that I mentioned, is a very highly regulated process in practice. Again, you don't need to know all of the details, but it's going to be relevant when we come back to talk about the PYC trial in a couple of slides' time. But mRNA, which we use to form the proteins, and in this case, the polycystin 1 protein, which is most important in the major cause of autosomal dominant polycystic kidney disease, has, and I don't know if you can see my arrow on the screen here, but it has the protein part of the RNA, which is what makes the actual protein, but it has these regulatory sides on it, this what's called a 5' untranslated region and a 3' downstream untranslated region. And this area helps regulate whether that gene is switched on or not, and how much of the gene is expressed. And again, just a sort of little schematic I made up last night, just demonstrating that there are some of these regulatory factors. This is one that's targeted by some therapeutics out there at the moment that affects whether this is switched off or not, and therefore determines how much of the gene is actually going to be turned into protein, which again is central to the problem for the condition. Genes we've touched on, about 85% of cases are polycystin 1 mutations, about 15% are polycystin 2 mutations, and then there are the handful of other genes. And we're increasingly interested in some of these genes because we're now starting to develop larger case series of families that have these different genetic mutations, and we know that they start to behave differently. So we know that if you happen to have, for example, the intraflagellar transport 140 mutation, that actually your risks of progressing to end-stage kidney disease are actually much, much lower than a lot of the other groups of patients. So as a clinician, where sequencing is getting done, it's now starting to provide us additional information that's useful for prognosis, and increasingly will become relevant for therapeutics that are targeted to particular aspects of the genetic abnormalities that are driving polycystic kidney disease. Almost at the end of the biology bits for you before we get onto the trial itself. But with this autosomal dominant story of this one faulty gene, this now for polycystic kidney disease is thought to be one of these what we call haploinsufficiency conditions, where the idea is that the one gene that you have left is just not enough for you to make a functional protein to stop the disease from forming. Polycystic kidney disease is actually quite interesting in this note, and we might come to that in the chat, that if you have too much of the protein, you also develop a disease. So there's a kind of Goldilocks zone that's the sweet spot. And the idea here is that as you fall below a particular threshold, you start to develop these cysts, and the less of the protein that you have, the more rapidly that those cysts will develop. And there is, again, a bit of data there to explain why that might be the case and the evidence that we have to support that. I don't need you to take in all the details here. What I want you to see basically is there are some blue things and there are some yellow things. So this is what happens in polycystic kidney disease. One gene goes wrong, polycystin 1, which, if you're interested, is sitting way up here on the picture. And then a whole lot of downstream activity starts to become dysregulated. And when people have looked at these different genes and proteins, these are thousands that tend to get dysregulated. Some of these, you'll notice, tend to be now the targets for some of the therapeutics that are out there. And I guess the critical part for me to stress here is that a lot of these are not treating the cause of the condition, but they're really targeting some of the downstream dysfunction that's caused because of the genetic abnormality. And in general, things that are switched on, we try and switch off. Things that are switched down, we would try and switch on. And we have a variety of different mechanisms, medications, and therapeutics to try and do that. There are a few challenges in terms of the targeting for these different options for therapeutics. So one of the questions that came through was about DNA. Can we just try and get rid of this gene? And that's maybe something in the distant future that might be available to us. And you may see a lot in the literature about some of the precision genetic tools that are available now. In particular, a technique called CRISPR-Cas that allows you to cut out quite precise segments of DNA and put them back in with the right bits of DNA. The problem for that is you have to be able to deliver it to the right sets of cells. If it's in a sort of dividing tissue, those cells will be sloughed off and you'd have to keep delivering it somehow. You have to make sure it delivers exactly to the right spot all the time, which again, current technology isn't great at doing reliably. And as I talked about earlier, if you give too much of the gene that gets switched on for various reasons, you'll also drive a disease phenotype here. So although that might be the distant future for therapeutics, it's certainly not where we are in at least the near future. In the third group, protein targets, that's what most of the additional therapies that in clinical trial at the moment, they're targeting those pathways that have been switched on or switched off downstream of the abnormality of polycystin within the cells. And again, there are some challenges on that. One, they tend to be affecting just individual pathways out of that huge constellation of issues that have been affected. And the targeting of some of those is challenging, and you get what's known as off-target effects. So we see that in a number of different therapeutic areas where it doesn't hit just the protein you're interested in, but unfortunately other areas too. So the drugs can be a little bit dirty in that setting or have unintended consequences. And the RNA therapeutics in the middle, I will talk about a bit more because that's how the PCY003 trial is focused. Just for completeness, I've just pulled last night. So here are some of the current therapies out there. So if anyone's got a particular question that they want to put into the chat regarding any of these, I'm very happy to address in more detail. And the link there for clinicaltrials.gov, if you're not aware about it, it's a great website. It's the American Clinical Trials listings. So you can type in polycystic kidney disease and it keeps up to date with trials that are active or recruiting or under exploration or closed and due to report. So a good resource for you there as well. One final bit before I move on to the PYC drug directly is that it's important, I guess, that we also think about what we're trying to measure in terms of our clinical trials. This is a nice little report that's just in press at the moment from colleagues in Queensland, just reminding us that it's great for us to be looking at total kidney volume over time and to changes in kidney function, but there are a whole wider dynamic of issues that people with polycystic kidney disease can suffer from or be affected by, and it's important that these things are incorporated into the clinical trials. And as patients, it's important that you advocate for these things to be part of the studies and things that we are looking at doing. Final point on just the background is doing clinical trials and getting from the idea of something through to the actual delivery is not a quick process. So the average is about 10.5 years for most therapeutics, with a massive investment to get to that point and a lot of things not making it along the way. So again, just unfortunately, I know there's a desire to get answers quickly, but these things unfortunately take quite a period of time while we make sure that what we're doing is actually safe. So with that, by way of background, and hopefully I'm keeping vaguely to time at the moment, I'm going to talk to you more specifically about the PYC-003 trial. So just again, back to the biology bit that I gave you to start with, patients who don't have polycystic kidney disease have two perfectly good copies of the PKD1 gene that makes polycystin 1, and therefore they make a normal amount of protein and have normal kidneys. In people with polycystic kidney disease, the autosomal dominant version, one of these proteins is not working normally. Therefore, the total amount of protein available is less than it should be. And that leads to all of the downstream dysfunction that we saw in those earlier slides. So the balance is lost. So the approach taken by PYC, which is fairly unique in this setting, but they have demonstrated it in other disease groups, so not polycystic or kidney diseases, but in some other genetic conditions, is to say, rather than trying to fix the broken gene by trying to do the DNA repairs and the complexities I mentioned earlier, what if we could improve the amount of the good gene that's already there that is expressed to try and restore the protein levels back up to a level that will stop or retard the disease from occurring? And that's exactly what this PYC drug is designed to do. So if you remember the earlier picture I put up of this regulatory element at the back end of the gene, it binds to that to try and stabilize this so that you can create more protein from the good gene, even though the bad gene is unfortunately still present. And this is where we are at the moment. So the rather unfortunately phased SAD study, so this stands for single ascending drug dosing study, with a view to then moving on to the multiple ascending dose study, and then along that same pathway I showed you before, a regulatory study where we get bigger patient groups through. So phase 1 studies, just to be clear, are about checking that it's safe. These are first in human sets of studies, and to start to explore some of the other endpoints that we'd like to see in clinical trials in terms of the efficacy of the agents. Where we are at this point in time, as of yesterday, we've finished the dosing in the sets of healthy volunteers, and we've started to dose into sets of patients with polycystic kidney disease at the lower ends of the dosing range that we think is going to be effective for people. The good news to date is that there have been no unexpected adverse reactions noted from the medication. We're not seeing any changes in kidney function that might suggest any toxicity from the drug. It uses some special technology to try and ensure that it's preferentially taken up in kidney cells where we want it to be delivered to. So one of the things to be concerned about would be whether there's any toxicity, and thankfully there's no evidence of any of that to date. No evidence of any injury. And the only part that we would like to change going forwards is that patients who were coming in for the study were required to fast, and quite a few people got headaches, which we had to report, which we think was probably all due to caffeine withdrawal, which I can certainly sympathize with. So hopefully that'll be removed from future protocols. The study is ongoing, and hopefully we will be able to ramp up the numbers across Australia. It's great that this is a therapy that is coming out of Australia for a disease, and that's pretty rare, just given the complexities and challenges with getting a therapeutic trial up off the ground. Again, fantastic if your sites are interested in being involved. And I believe even where your sites or sites no close to you may not be able to be involved, I think certainly looking at being able to get patients to those sites for the purposes of being part of the study if it's something that they are interested in. Again, just by way of a quick look at the eligibility criteria. So 18 to 60 years of age, so adult patients with polycystic kidney disease. The particular way that the polycystin 1 drug that the PYC agent works on acts means that we need people to have that particular mutation. So people who haven't been sequencing is done as part of the eligibility criteria. And there are a variety of other details, which again, you can find through the study team, or you can find out through clinicaltrials.gov, where all of that is listed. And with that, I will bring my presentation to an end, and I'm very happy to answer any questions that might be there.
Fantastic, thank you. Excellent. So we've had, I think, what I was hoping for this webinar was we've got a full lay of the land from the beginning of genetic testing to being on a registry, how that can be used for trials, and now an actual current trial that is happening. So off the back of that, I think we will just start with you, Aron. If someone wants to join the next phase of the PYC trial, how do they go about practically doing that?
Yeah, so on those slides, which I think you're going to circulate afterwards, Jess, or there'll be access to. So there's a link, the second last slide, that's got the QR code, and it's got a couple of email addresses that will link into telling people the sites that are open near them. It also lists the investigators that they can contact as well if they wanted to approach them directly, whichever state they're in, or if they don't happen to have somebody in their state, then they can reach out to the generic email, and then we can try and link things up.
Fantastic, thank you. And yeah, we've got a few comments here saying some people are interested in joining, as well as the registry. While I'm chatting, anyone listening, feel free to pop your questions in that Q&A. We are using it. In the meantime, I'm going to go through some of the questions that were sent through earlier as part of the registration process. So I actually wanted to go back to you, Jono. There was a question about the registry and specifically data sharing. Is there genetic testing results shared between Australia and the UK? And if not, why not?
Yeah, very good question. We get asked that a lot, actually. It's sort of a can of worms that we don't want to open, particularly when it comes to data sharing internationally. And I think I would say the one sole reason is that there's such a huge difference and difficulty between the different ethics boards of certain countries. So, as well as who actually owns the data. So, from a registry point of view, that would be our current institution, so that's Garvan. And if there is data sharing of genomic data, it would need to be within a, I guess, mutually collaborated, secured environment. There were talks to explore that particular area, but it will take couple more months or so before we can realistically understand what we can and can't do on an international basis.
And then more locally, within Australia, if you have, I'm not sure if this is the case, but if you have been tested elsewhere or you've already been entered with your diagnosis, is that data that you're able to consent to give the Garvan, or does the testing process have to be like through you?
Yep. So we actually do both, actually. So if participants have had genetic testing elsewhere, and they've consented to share that data with us, we're able to collect and store that securely within our current servers. Otherwise, if they are undiagnosed, we have the means to conduct genome sequencing on our end as well.
Fantastic. Thank you. The next question I have, I think maybe Ella, you'd be best placed to answer. And that is, you may have briefly covered this, but when should you test? When is the best time, or when do you see most people come in to get genetic testing, like age-wise?
It's really, really dependent. We have a lot of people come in for family planning purposes. And then we've been getting more and more because we know that there's a Medicare item number, so it's becoming a lot easier for people to get testing. And then the other thing we are saying is, it does there are some trials, not for PKD, but other things we know we have to have a genetic diagnosis to be eligible for that trial. So, if anything, I'm sure Aron can answer more about this, but we do know that sometimes it's better to have something sitting and ready to go rather than waiting, 8 months for a genetic diagnosis. But people come in at all times. There's so many different reasons. It's really just what's going to be best for them.
Awesome. Actually, I personally was just going to ask, knowing that the testing may take that long, but if people are signing up for clinical trials that have a set time, say, how does that work? Is it fast tracked or is it fast tracked essentially for a clinical trial? Your diagnosis?
I can answer some of that bit to say that yes, for our part at the moment, they have been able to negotiate expedited testing for the polycystic kidney disease genes for exactly that purpose, so that people will be able to enroll in the trial or find out if they're suitable.
Okay, awesome. Thank you.
It's
We
always-
one more
sorry.
question. Oh, sorry, Ella, no, you go.
All I was going to say is it's always worth, if you are getting genetic testing, just ask about fast tracking, because sometimes we also fast track if people are like, I want to have a pregnancy in like 3 weeks' time. We're like, okay, can't get you in 3 weeks, but we can fast track that result. So that sometimes there are other reasons that we fast track results.
Right. Okay, that's great to know. Thank you. Just relating again to PYC, again, this information was likely to be online, but Aron, if you can answer, are there plans to roll out this trial in Brisbane or Queensland in the near term?
Yeah, so there's I think a site in Queensland that's being explored at the moment. I think I was trying to remember where exactly. I'll have to double check and can try and post that to you afterwards. Unless there's somebody else online who knows and can answer for me. But yes, so look, we're enthusiastic for as many sites to get involved. This is still obviously the early single ascending phase. And then as we get to the multi-dosing, when you look at the numbers of participants, we kind of need hundreds of patients to start to answer those sets of questions. So we'd really like as many sites to be available. Neve's just come to say, yeah, two Queensland sites in Brisbane and Gold Coast. There you go.
Fantastic. Thank you, Vanessa. I saw a name pop up earlier, part of the PYC team. Okay, thank you. I am going to go to another question. Oh, actually, a lot of these, you'll be glad to know, have been answered, I think, during the course of the webinar. A question for somebody. For the PKD diagnosis, obviously it's gene related, but can the gene be carried but then not actually develop into the disease?
I'm happy to answer that. So it's dependent on the gene. What we know with PKD1, it's pretty, we call it penetrant, and there is some variability, obviously, within, like, you might see your family member with a slightly different presentation, but we do know with PKD1 specifically and PKD2, you will get cysts in your kidneys. There are other genetic conditions that are incredibly variable, where someone might get the condition, others don't, but with PKD, ADPKD specifically, and those two genes, we do know that it's pretty, if you have a gene change, you will develop cysts in your kidneys, but we don't, it's very dependent on you, what that future might look like, and that's different for every single person.
Mm-hmm.
I answered it.
Thank you for that answer. Similar question, and again, this is getting down to the science and the mum and the dad and the gene. Can it skip a generation if you had someone that, I guess, gets both recessive genes or doesn't inherit it? Can it skip?
Yeah, sorry, I can take that one again. So with with dominant inheritance, we know that if it's passed on, if it's not passed on and that child does not get it, there's no skipping generations for dominant polycystic kidney disease. With recessive, that person, if they have both copies knocked out, if they go to have a child, their partner would also have to have be a carrier. So with recessive conditions, it's not really skipping generations, it's more just seeing if that partner is also a carrier. But we do know that the carrier frequency is quite low. So most people who have autosomal recessive PKD and go on to have children will often get their partner tested. And it's usually a low risk, but it's always something that we discuss with them. That makes sense.
Yeah, absolutely. Thank you, Ella. another interesting question is it possible to remove the gene in utero? Yeah, is it possible to remove it in utero before a baby is born?
Don't know. I might let Aron talk. Well, he's answered that you can't get rid of a gene, but at the moment there are no fetal therapies for PKD. Unfortunately, the only thing you can do is go through the IVF process, and that's before obviously even implanting that embryo into the uterus. But there are currently no, because we can't get rid of the gene, and there's probably something way in the future. I don't know if Aron, you agree.
No, I completely agree. And there's an interesting story here as well, in that we did a project, consumer outreach, must be a decade or so ago now, asking people with polycystic kidney disease around, okay, if you found out that you, as a hypothetical, that you had passed on polycystic kidney disease to your child in utero, would you do anything about it? And actually, it was a pretty low frequency that would actually want to do anything, and the risks that would go with that. It was actually around about 6 or 7% of respondents said that would be something that was on their radar that they would want to do, despite this being a group of people with a fairly significant condition. And that was a decade ago. And technology's moved on a lot. Our options and therapies, not even the specific ones, but things that slow down progression of kidney disease, have evolved dramatically over that period of time. So again, if there is anyone on board online who's kind of concerned about this, I think from where we were a decade ago, the future's pretty bright in terms of opportunities and therapeutics, and where we're heading. It's moving very fast.
Very exciting. On that, I think, Aron, with your years of experience, would you say that more clinical trials are moving towards the need for genetic testing and a genetic basis, or not?
Not all of them. So those sets of trials that are targeting some of those downstream proteins and things that are not working as a result of the mutation, those are largely agnostic to the underlying genetic cause. They are downstream effects, and the hope would be some of those therapies would be of some benefit no matter what the mutation will be. But then there are other therapies like the PYC trial, which are absolutely targeted. That the way it works is only going to work if you have that polycystin 1 mutation. So we will probably start to see, as more of these targeted therapies come out there, that genetic diagnosis will be more useful. And again, I'll also just re-highlight the fact that it's also useful from a prognostic perspective, as it starts to, the guidelines that came out from our the kidney disease, the global body, earlier this year now, is sort of increasingly talking about the use of ADPKD and the gene name where sequencing has been done, and we have a genetic diagnosis, because it can help to inform prognosis and then for treatment options as a result. So I think we're going to see more and more of it, and we will see targeted therapeutics in some specific instances.
Excellent. Well then, Jono, I think your inbox will soon be flooded. He's hoping that we'll have more and more people sign up for the registry to facilitate exactly that. Another question has come through, saying, can a person's gender, this is potentially a little unrelated, but can a person's gender determine the aggressiveness of the disease?
From an autosomal dominant perspective, that's the same genetic change no matter what. I'm not sure if there are small intricacies. I'll let Aron answer that, but from my perspective, is it genetic change? It is the same genetic change, male or female.
Mm-hmm.
Yeah, this is a really fascinating set of questions. So absolutely. So the underlying gene change is the same, but how that expresses itself definitely changes in certain circumstances. And we don't really understand what all of those factors are at the moment. And that comes back down to some of that clever stuff around the homeostasis and control mechanisms that sit there. Why do some people progress really rapidly? Why do some people not? For example, I look after two brothers who are both have polycystic kidney disease from their father. One of them is absolutely fine in his early 30s. His brother had his transplant in his mid-20s. So the 32-year-old has impaired kidney function, but he's still quite a long way away from transplant, yet they have exactly the same genetic mutation. They're just a few years different. They've grown up in the same environment. So there are these funny factors in there that we don't fully understand yet.
Okay, more research to be done. Always. Well, oh, I think another question has come through. Okay. With genetic understanding moving fast, would it be advisable to have another genetic test? And if so, like how many years? Because this person had the last one 3 years ago and showed a variant of uncertain significance.
Yeah, I would, whoever did that testing, I always recommend every few years to just reach out and say, look, is there value redoing this testing? It will be based on the clinician that did your test. Sometimes there's all these different, some people say more, some people say less, but basically it will be up to the clinician to decide whether there's value redoing that test. But it is a good idea just to reach out every few years.
And even if you already have received your diagnosis, that stays. There's no need to retest.
I can see if it's a variant of uncertain significance. If it's a positive result, then that's good. We've got the answer. But if it's a negative result or a variant of uncertain significance, there is value. There can be value redoing those tests in a few years' time. So it's best to just discuss it with your clinician because they'll be able to give you a guide on how many years or if there is further testing available to you.
Excellent. It's the value of a genetic counsellor. Thank you. This one might also be for you, Ella. This is a question we have had quite a few times, but I think it's worth chatting through every time. This person has recently been diagnosed and her children are 18 and 16. So what is a good age to discuss PKD with your children?
So it's really up to you, and it's obviously a very personal discussion. But again, I will say genetic counsellors, we speak to children and we speak to teenagers all the time about genetic conditions. So if we do know, it could be a good time to have that conversation with them if you feel ready, but also know that you could ask for a referral to a genetic counsellor just to actually begin having that conversation and feel a bit more supported throughout that conversation. And we know that sometimes if you make it a nice, friendly, open conversation, it sounds less scary. And sometimes the more that they hear about it and the more that kids normalize it in their head, it becomes a less scary thing. It just goes, okay, this is what it is. This is what I have to do. But we're always happy to chat to teens and kids about any genetic diagnosis.
Fantastic. Yeah, I think for this person listening, I think reach out to a genetic counsellor. I can give you Ella's details as well. And also we have community and we have people who have been in the same boat that you are in. And even if you just want to chat to someone else who has been in that situation and ask, how did you tell your kids about it? I have no doubt that, I can connect you with other community members as well. So thank you. And on that note, we are at time. So I'm going to wrap up the question time. Thank you so much, the three of you, for your presentations. I think they fit really nicely together, giving us just that tools in our belt of all the different aspects of genetic testing and diagnosis and how registry can actually be really beneficial, particularly to Australia and research and the new clinical trials that are underway. So there's a lot of information, but extremely helpful and I think really practical. So thank you for taking time out of your evening. Thank you everybody for joining us. Again, a lot of this information is on our social media or our newsletter. And if you have any questions, feel free to email me and I can connect you with who you need to be connected with. And on that note, have a great evening and we'll see you next time. Thank you.
Thank you so much.